Hi, my name is Megan Rokup, and I work here on MIT campus at the Broad Institute of MIT and Harvard, which is a biomedical research institute. Today, we're going to do a lesson talking about using DNA to identify people. And as you can see on this slide, I'm showing you a picture of DNA. This is what DNA looks like. It's a molecule made up of atoms. There's a lot of carbons and hydrogens and nitrogens and oxygens. And it forms a three-dimensional structure shown here as a double helix. Now, the backbone of this structure, which is the two spirals you can see, are always the same along the DNA molecule. But the rungs in the middle can be different. And there's actually 4 different kinds of rungs, and we abbreviate them A, T, G, and C. So while this is what DNA actually looks like, a three-dimensional molecule, on this slide you can see how we represent DNA. We represent DNA by writing out the sequence as a series of A's, T's, G's, and C's. Now, this slide shows about 2,000 letters, and your first instinct may be that this looks like a lot of DNA. But actually, each cell in the human body contains 3 billion of DNA letters that you get from your mom and 3 billion DNA letters that you get from your dad. So this is only 2,000 letters. Imagine the amount of DNA in one of your cells. I want to ask you a question about this slide now, which is the following. Let's say that this 2,000 letters of DNA was the sequence of DNA at one particular region of a chromosome in me. And these are the series of letters in the sequence and the order that you would find them at some position in my DNA. If we looked at the same position in the DNA in anyone else on Earth, how many of these 2,000 letters do you think would be different between me and anyone else on Earth? I'm going to give you a minute to discuss that, and we'll answer the question when you get back. So let's say this screen is 2,000 letters of DNA in some position in my DNA. And the question was, how many of these letters of DNA would be different between me and anyone else on Earth? Well, the answer is maybe 1 or 2. So let me show you an example on this slide. We've got our 2,000 letters, and maybe in me, this whole thing is the order of the letters that you would find in the order and the sequence. But maybe in someone else in the world, this T right here might be a C, and maybe down here, this A might be a G. All the other letters would be the same sequence in the same order. And that's because all humans on Earth are at least 99.9% genetically identical to every other person on Earth. So we're really similar. Now, everyone on Earth has a different DNA sequence than everyone else on Earth, except for one exception. I can say that I have different DNA than anyone on Earth, but the one exception would be if I had an identical twin. 2 identical twins have the identical sequence of all 3 billion letters that they get from their mom and all 3 billion letters that they get from their dad. But everyone other than identical twins have different sequences from each other. And so we can use DNA sequence to tell people apart. And this is a technique called DNA fingerprinting or genotyping. So on my next slide, I'm going to show you those 2 words. This is what our lesson's going to be dealing with today. It's one technique, it just has 2 different names. It's a technique to use DNA to identify people. The reason it's called DNA fingerprinting is that that's an analogy to actual fingerprinting, where every person has a different pattern of bands on their fingertips. So every person also has a different DNA sequence. So we can basically fingerprint people by using their DNA. The other term is genotyping, and this is because this technique allows us to see what type of genes people have. So I told you that different people have different DNA sequences, and let me show you what I mean by that on my next slide. Before I show you 2 different people differing from each other, first I just want to show you the DNA sequence from one person. And I'm showing it to you in this format so that you can see how exactly we write out a representation of a 3-dimensional structure. So here's a sequence of DNA from a person. The double helix that you see is the 3-dimensional structure of DNA. But I can write out one strand and then write out the other strand next to each other using a series of letters. And that's what I've done on the right-hand side of the slide. These letters are A's, T's, G's, and C's. And anytime you see an A on one strand, you see a T on the other strand. Anytime you see a G on one strand, you see a C on the other strand. A's bond with T's and G's bond with C's. So this is how we write a DNA sequence. Now normally actually we don't write it vertically, so I'm going to turn it for you on my next slide so that it's horizontal. So here's an example of a DNA sequence from a person. I'm going to call them human number 1. When I say that people have differences in DNA sequence, what I mean is shown on this slide, where now I'm showing human number 2, and there's a position where human number 1 has a G on the top strand, and human number 2 at that same position has an A on the top strand. All the other letters are the same. Now you would actually have to look through over 1,000 letters of DNA to find this difference, but when you do find them, this is what they look like. So we use DNA fingerprinting to look at differences in DNA like this to tell people apart. And what I'd like to ask you to do now is to brainstorm uses of this technique. So in what situations do you think it would be useful to use the DNA of people to identify them? So the question was, what uses would you have for a technique where you could use the DNA of people to identify them? Remember, DNA fingerprinting or genotyping allows us to identify people based on their DNA. So what would we actually use this technique for? Well, hopefully you've had a chance to brainstorm, and on my next slide I'll show you some of the possibilities that I came up with. One situation is paternity testing, or trying to determine the parents of a child. Another situation is forensics. So let's say there's a crime scene and a hair or a piece of skin is found at the scene of the crime. You could isolate the DNA from that sample and figure out who the person was who left that sample there. And then my last example is one that actually we do here at the Broad Institute all the time. So what we do is we look at the patterns of DNA between people and compare them routinely. And the reason that we do this is to try to figure out which portions of the DNA are associated with specific diseases. So we take thousands of people who have a disease, let's say heart disease, and thousands of people who don't, and we look at the differences in their DNA sequences. We look for the differences that always look one way in the people with the disease and always look a different way in people who don't have the disease. And that's how we find regions of the genome that are associated with different diseases. But today in our lesson, we're going to focus on the first two uses in paternity testing and forensics. Now, when you're looking at genes that are associated with diseases, you often use the kinds of sequence differences that I showed you before. So on this slide, I'm reminding you that humans can differ by sequence, for instance, human number 1 having a G and human number 2 having an A. Although these are the differences we use for mapping genetic diseases, these are not usually the differences that we use when we're doing paternity testing or forensics. So let me show you what those look like. On my next slide, I'm showing you a different way that people can differ in their DNA. Before I was showing you a difference in sequence where a letter was different. But here, the sequence of these two people is the same. Both people have a repeat in their DNA that says GT, GT, GT over and over again. The difference is that human number 1 has 3 repeats. Their DNA says GT 3 times in a row, where human number 2 has 5 repeats, GT 5 times in a row. So these two people don't differ in sequence, they differ in length. And on my next slide, I'll show you that if you count up the letters of DNA in the top strand of each person's DNA, human number 1's DNA would be 18 letters long, and human number 2's DNA would be 22 letters long. So if we have a technique that separates DNA by size, we can actually tell these people apart. We do have a technique that does this, and I'll show you that on my next slide. This technique is called gel electrophoresis. And there's 3 things that I'd like you to remember about gel electrophoresis. The first is the main point, which is that it's a technique that separates DNA by size. The second is how it works. So you actually put the DNA of people into a slab of gel that feels a lot like Jell-O or gelatin. Now this gel is a matrix. And what I mean by that is I could turn this room into a matrix if I took a whole bunch of pieces of string. And every piece of string, I taped one end to that wall, and then I stretched each piece of string across in a different pattern and taped the other end to that wall. So imagine the room that you're sitting in now, full of string. That would be a matrix. Now if I wanted to race 2 molecules through the matrix, let's say I wanted to race a very large molecule and a very small molecule. They would move through the matrix with different speeds. This is what we do with DNA. And the reason we can get DNA to race through the matrix is that DNA is negatively charged. So, what would you do to the other end of the room if I were a piece of DNA to make me want to go to the other end of the room? So I'm going to give you a moment to think about two things. First of all, what would you do to the other end of the room to make me want to move there? And secondly, if there were two pieces of DNA here, one very large and one very small, how might they behave differently as they race towards the other end of the room? So if DNA moved through the matrix, first of all, why would it want to move in the first place? Well, if I'm DNA and I'm negatively charged, if you make the other side of the room positive, then I'll be attracted to the other side of the room and I'll want to move there. Now, what if there were two molecules of DNA and they both had to move through the matrix, but one was very large and one was very small? Well, the small one can move quickly through the matrix because it's small enough to fit in between the holes in between the pieces of string. Whereas the large molecule would try to move, but it would be very large, and so it would get tangled up and thus move slower. And that's how gel electrophoresis works. I'd like to show you how this technique works in my lab, but first I just want to introduce you to the principles, and then we'll go into lab to take a look at the technique. So, let's say that you had DNA samples from people, and you wanted to separate them by size using a gel. What would you do? Well, on my next slide, I'm going to show you the first step that you would do. What you would do is, you would take all the DNA in the sample, which remember, every human cell has 3 billion letters from their mom, 3 billion letters from the dad. And we're only usually analyzing a very small amount of that DNA. So what we do is we use a technique called PCR, where we pick a left and right boundary of the DNA, and we copy only the segment in between those boundaries. So on my next slide, I'll show you what that looks like. You pick the left and right boundaries, and then you make many, many copies of just that region of DNA. And here I'm showing you only a few copies, but in the lab we make millions and billions of copies. So now we have a bunch of copies of just the region we want to look at. So basically, here's how the technique works. You isolate the DNA from the person, you make many copies of just the region that you want to study, and then you're going to run the DNA through the gel. I'd like to show you what this actually looks like in lab, so why don't you come with me and we'll go do this technique together in my lab. Hi, welcome to my lab here at the Broad Institute. I'm going to be showing you how this process works here in lab, and here you can see the equipment that I'll be using to show you how to use gel electrophoresis to identify people based on their DNA. So, typically we store our DNA samples like this in these tubes, and I got these tubes out of the freezer. Typically we store DNA at minus 20 degrees Celsius, which is the temperature of a standard freezer. The first thing that I'm going to do is I'm going to take a little sample of those samples of DNA, and I'm going to put them into these smaller tubes, which we'll use in these smaller tubes to copy the segment of the DNA that we're interested in. So I'll be using my pipette to transfer, for each sample, a small amount of the liquid into the small tube. The next thing I'm going to do is I'm going to add the reagents or chemicals that I'll be needing for each copying reaction to each one of the tubes. This includes all of the necessary chemicals that take you from having a small amount of DNA to copying it into a large amount of DNA. Each time I pipette a different sample, I use a different pipette tip so that none of the samples are contaminated with each other. So now that I have all my samples in the tubes, at this step, I'll be adding the mixture of all the chemicals necessary to do the reaction. So I have that here on ice in this tube. The reason I have everything on ice is because this reaction is sensitive to temperature, and so I want to keep all the reactions on ice until I'm ready to start the reactions. Again, I'm changing pipette tips each time so that none of my samples contaminate one another. So now that I have all of my DNA samples mixed with the necessary chemicals, I'll be putting these smaller tubes into what we call a PCR machine. So remember, PCR is the name of the technique to copy a segment of DNA to make many copies of it. And so this is my PCR machine right here. And these small tubes, I'll just give them a little mix there, and they fit right into these holes inside the tube, inside the machine. So when I've put all 4 of my tubes into the machine, I just close the lid, lock it down, and then I hit start, and the program's going to start running. What the program is actually going to do is that this machine, although it's called a PCR machine, all it really is a heating block. This machine is a heat block that changes temperature really rapidly. So it's going to change temperatures, and each step of the copying reaction happens at a different ideal temperature. So as it changes the temperatures, then the steps of the copying reaction can occur in the order in which you want them to. Now remember what this machine is doing. This machine is doing the copying reaction that I'm showing on this slide, where we make many copies of just one piece of the DNA. I want to give you a moment to think about, why is it that this copying reaction is necessary? Why can we not just take all the DNA from a person and directly analyze it on a gel? I'm going to give you a moment to think about why the copying step is necessary. The reason we need to copy the DNA is twofold. The first is just an amount issue. The amount of DNA we can typically isolate from a person simply isn't enough to see it directly on a gel. Instead, we need to copy it so that we have more DNA such that we are able to see it on the gel. The second reason is that we only want to look at one segment of the DNA. We don't want to look at all the DNA from the person. Therefore, the copying reaction allows us to just select one piece of the DNA from that person, make many copies of it, and then visualize just that segment on the gel. Now, this process actually takes a few hours, and so I set up PCR reactions before we started filming, and I have them in this ice bucket here. So the next thing I'm going to do when the PCR reactions are done and I have my segment copied is I'm going to transfer, again, a small amount of that into these tubes right here. And these tubes, I'm going to mix the DNA with an orange dye. This dye is going to allow me to see the DNA, and then I'll be loading it onto this gel here. So the first thing I'll do is transfer a small amount of orange dye into each of the tubes that I'll be placing the DNA samples into. You can see this orange dye in my pipette tip. It's just a colored substance that we can see with our eyes. DNA is transparent, so we can't see it with our eyes. Okay, so now I'm going to be transferring a small amount of each sample into those tubes. So here's my PCR reaction. I place it in the tube to mix with the orange so that now we can see it. Okay, so now that we have all of our DNA samples to load onto the gel, I'm going to load them onto the gel. The way that works is that this is your slab of gel right here. If you can see from the side, the gel is actually coated in 2 pieces of plastic, but the opaque substance in the middle is the gel matrix. Now this gel right now has a plastic comb in it that's protecting 12 holes in the gel. We can load each DNA sample into one of these holes. So I'm going to put the gel down onto the lab bench. And for each one of my DNA samples, I'm going to load them into one hole in the well. In the gel, sorry. So I'm loading my DNA samples of the different people individually into each hole in the gel. And the orange dye helps me to see that I've actually successfully gotten the DNA into the hole. And once I've loaded all the DNA samples, I'm actually going to load one other special sample. First, I'm going to add a little DNA orange dye to it. This special sample is not the DNA from a person, but rather it's a mixture of samples of DNA that we buy from a company. And the different pieces of DNA in this mixture are pieces of DNA whose sizes we know. And so we use it like a ruler or a standard to be able to tell the lengths of the pieces of DNA that we don't know in the samples from the people. So this will be our ruler or standard, and we call it ladder. So now what I'm going to do is I'm going to hit this button, and that starts the current running through the gel. So now the DNA samples are going to move from the holes where we loaded them all the way down to the end of the gel right here. And so you'll see the orange dye progressing. The orange dye, remember, is not actually the DNA. The DNA is transparent and you can't see it at this step. But the orange dye allows us to see that the samples are traveling. Once the orange dye reaches the end of the gel, we'll expose this gel to a certain wavelength of light, and that will allow us to see the DNA. This takes about half an hour, and so I actually started another gel for you before we started filming this segment. And you can see that the orange dye is almost at the end of the gel here. So this gel is almost done running. And when it's done running, it'll beep to tell me it's done, and then we're going to take it into the darkroom so that you can see the results. So here we are at the darkroom. This is where we're going to visualize the gel. I've got my gel here, and we're going to go in and shine a certain wavelength of light on it so we can see the DNA. So here we are in the darkroom, and I've got my gel. I put my gel into this light box right here, and then I simply turn on the light, and all of the bands of DNA appear. So here you can see the results of the gel. Lane 5 is not a sample from a person. Remember, that's ladder that we bought from a company. So you see a series of bands of DNA. Each one is a different size, and we know the sizes of those DNA. So that's our standard. Lanes 6, 7, 8, and 9 contain the DNA from 4 different samples. For instance, in lane 9, you can see that there's a single band, whereas in lane 7, there are multiple bands. So what I'd like you to do now is take a moment to discuss why you think it is that some people show one band, whereas other people show multiple bands. I'd like you to write down for each person how many bands that you see, whether those bands are longer or shorter, and then also explain why some people show multiple bands and some people show one band. When we were in lab, you saw a picture of a gel that looks a lot like this slide here. As you can see in this slide, some people show one band of DNA, other people show 2 bands of DNA. Well, why is that? The reason is that people who only show one band of DNA actually have double that amount of DNA, as you can see in the difference in the intensity of the band. Remember, everyone gets their DNA from their parents, and everyone gets half their DNA from their mom and half their DNA from their dad. So if a person shows only one band in this gel, that means that the DNA they got from their mom was the same length as the DNA they got from their dad. So there's 2 times as much DNA in that band, which is why it's darker. A person who shows 2 bands, however, must have gotten a different length of DNA at that section from one parent than they did from the other. And so you see 2 different lengths of DNA in their lanes. So that's how you interpret a gel like this. In this gel, for instance, as you can see from the ladder markings on the left, remember the ladder is like a ruler for us, person number 3 has about 22 letters long, whereas person number 1 and person number 2 have DNA that's both 22 letters long and 18 letters long. So I just want to show you a quick pictorial view on this next slide to remind you that when you're looking at these gels, you're looking at the DNA that comes from the parents. And here's a picture of the DNA actually sitting in a human cell. The round part is the nucleus, and as you can see, there are chromosomes in the nucleus. And in my picture here, I'm only showing 3 chromosomes, a very long one, a medium one, and a short one. In actual human cells, there are 23 different chromosomes. you would get 23 chromosomes from your mom and 23 chromosomes from your dad. So here on this slide, I've labeled a section of DNA in the same position of the long chromosome from the father and the long chromosome from a mother. And as you can see, one section has 3 repeats of CCT, while the other section only has 1 region of CCT. Thus, this region from the 2 parents of this person is different sizes, so this person would show 2 bands at this position. So now let's go back and look at the gel I was just showing you. I've now labeled the lanes a little bit differently. I've labeled one person as me, another person as the person I refer to as mom, and another person as the person I refer to as dad. And I have 2 questions for you. Using the data in this gel, can these people be my parents? And my second question is, are these people my parents? So I want you to discuss as a group whether you think these people can be my parents based on this gel, and whether this gel shows you that these people are my parents. And we'll discuss